everyone. Welcome to an online webinar on medical device and trials. Hi, I am Dr. Shivangi Singh, Program Director, FINENESS Institute of Clinical Research, Delhi. Firstly, we'll look into what is a medical device. A medical device is defined as a medical tool which does not achieve its primary intended action in or on the human body by pharmacological, immunological, or metabolic means, but which may be assisted in its intended function by such means. Anything used for the therapeutic and or on diagnostic purpose in humans, which is not a drug, means that it is not dependent on being metabolized for the achievement of any of its primary intended purposes. It may be an instrument, apparatus, appliance, implant, material, or any other article, whether used alone or in combination, including a software or an accessory, intended by its manufacturer to be used specially for human beings or animals for one or more of the specific purpose of detection, diagnosis, prevention, monitoring, treatment, or alleviation of any physiological condition or state of health or illness, replacement or modification or support of the anatomy or congenital deformity, supporting or sustaining life, disinfection of medical device, and control of conception. Now we'll look how a medical device is different from a drug. There are different parameters. The first one is use. Drug is used in vivo use inside the body. Device is in vivo or ex vivo, that is outside the body. Active components are based on chemistry and pharmacology. In device, it is based on mechanical, electrical, and material engineering. Diagnostic or therapeutic intended use. Drugs are used for therapeutic intended use, and device is for diagnostic or therapeutic intended uses. Quality depends on safety and efficacy. In device, depends on safety and performance. Trials, it is done in clinical trial. And devices, it's known as clinical investigation, or it is known as clinical evaluation. Manufacturing requirement, that is good manufacturing practice, GMP for drugs, and for device, it's quality management system or QMC. Animal toxicity, local and systemic toxicity is there. In device, it is biocompatibility. Life cycle, long product life cycle is for the drugs. Devices is comparatively short product life cycle. Sample size is larger in drugs, large population is taken, and device, limited population. Medical device includes everything from a highly sophisticated computerized medical equipment like MRI machine down to a simple tongue depressor. Other examples of medical devices include orthopedic implants, pacemaker, cochlear implants, catheter, blood glucose meter, pregnancy test kits, perfusion sets, sutures, etc. They can be temporary, permanent, used by professional, used by a person without supervision. For example, temporary use, cervical collar, walking stick during an injury. Permanent use is cardiac stent or orthopedic implants. Professional use is blood assay monitor. We'll just look at the common types of medical devices like ablation device, bone cement, cardiac stents, catheter, disposable hypodermic needles and syringe, disposable perfusion sets, internal prosthetic replacement, IV cannula, orthopedic implants, surgical dressings. Now we'll look into each of it one by one. Ablation is removal of a biological material from human organs by vaporization, cryotreatment, radiation procedures, chipping, or other erosive processes. In medical sector, ablation is performed usually through surgical methods for the removal of biological substances, usually a small tumor. Bone cement, as shown in the image, can be defined as a family of material that consist of a powder phase and a liquid phase, which after mixing form a plastic paste which have the ability to self-set once implanted in the body. Cardiac stent are expandable coils made of metal mesh, as it's quite visible in the image. This device is designed to support your artery walls, keep your artery open, and improve blood flow to your heart. Catheter is a soft hollow tube which is passed into the bladder to drain urine. Catheters are sometimes necessary for people who, for a variety of reasons, cannot pass their bladder in the usual way, passing urine in the toilet or urinal. Disposable hypodermic needles or syringe. A hypodermic, hypo meaning under, and dermic the skin, needle is a hollow needle commonly used with a syringe to inject substance in the body or extract fluid from it. They may also use to take liquid samples from the body, for example, taking blood from a vein in a venipuncture. Disposable perfusion set used during cardiopulmonary bypass to connect multiple coronary cannula to a single infusion line. Internal prosthetic replacement, an artificial substitute or replacement of a part of a body, for example, tooth, eye, facial bones, palate, hip, knee, and other joints, the leg or an arm. Many uses. IV cannula is intravenous cannulation is a process by which a small plastic tube, a cannula, is inserted in a peripheral vein. The subsequent venous access can be used for administration of fluids, medication, and nutrition. In some cases, blood samples can also be obtained from the cannula. Orthopedic implants. Orthopedic implants are those material that are used for hard tissue application to replace bones and joints. Also included in this category are fixation plates that are implanted to stabilize fractured bone. Surgical dressing. A dressing is a sterile pad that is applied to a wound to promote healing and protect the wound from further harm. A dressing is designed to be in direct contact with the wound as distinguished from the bandage, which is often used to hold the dressing in place. We look at the how medical devices are classified. Uh, the risk-based classification is as is low is class A, low to moderate class B, moderate to high is class C, and high is class D. Some example to get a better understanding. Class A, for example, surgical dressing like absorbent cotton wool, nasopharyngeal catheter. Class B is hypodermic needles like the ones used by the dentist. Surgical dressings like liquid bandage. Class C, catheter like urethral stent. Disposable perfusion sets, hemoconcentrator set. Class D is catheter like angiographic guide wire or a cardiac stent or a coronary stent. Now we'll look at the regulatory framework in India and different countries, how they are approved by the government. In India, medical devices are regulated as drugs. This sector is very small by size as compared to the rest of the manufacturing industry. Medical device imports supplies around 77% of the market. The Indian market is growing at a CAGR, compound annual growth rate, of 15%, making India one of the fastest growing market in the world. Strong drivers for this robust growth of medical technology industry exist in India. In order to have a specific requirement for medical device, Medical Device Rules 2017 has been published. These rules are framed under the existing Drug and Cosmetic Act 1940, the requirement for import, manufacture, clinical investigation, sale and distribution of regulated medical device has been prescribed in Medical Device Rule 2017. These rules have been implemented from 1st Jan 2018. Another important point that you need to know is Global Harmonization Task Force, GHTF. GHTF was founded in 1993 by the government and industry representative of Australia, Canada, Japan, and the European Union and the United States of America. The purpose of the GHTF is to encourage a convergence in the standard and regulatory practices related to the safety, performance, and quality of medical devices. The GHTF also promotes technological innovations and facilitates international trade. The primary means by which its goals are accomplished is by the publication and dissemination of harmonized guidelines documents for basic regulatory practices. Most developed countries have inadequate national policies and regulation covering medical devices. The WHO collaboration with the GHTF could facilitate access for developing countries, both those importing and those wishing to manufacture too. Information on the major regulatory system for medical devices, device approval and health technology assessment from highly regulated market, adoption of a single medical device nomenclature, innovative technology advances, network for post-market surveillance and vigilance. Uh, Now we'll look at the classification that is being used worldwide. In India, it's low is A, low to moderate B, moderate to high C, and high is D, same as GHTF. In Australia, low is 1, low to moderate is 2A, moderate to high is 2B, and high is 3. In Canada, it is low is 1, low to moderate is 2, moderate to high is 3, and high is 4. In Europe, it is 1 is low, 2A is low to moderate, 2B is moderate to high, and 3 is high. In Japan, low is general, low to moderate is controlled, and moderate to high and high are highly controlled. In US, it is low is 1, low to moderate is 2, and high is 3. Worldwide medical device trial. Regulations of the US is Federal Food and Drug Cosmetic Act. In Europe is Medical Device Directive, MDD. Pre-market and post-market supervision is done by FDA. The Center for Device and Regulatory Health is accountable. For Europe, it is a notified body having authority to grant CE certification. The risk, as already been discussed, is 1, 2, and 3. In Europe is 1, 2, 2A, 2B, and 3. Approval system is general control for 1, 2 is pre-market notification process, 3 is special control. And for Europe, manufacturer need to exhibit CE marking in order to ensure the device is safe and fit for its intended use. For Japan and Australia and Canada, it is regulated under Pharmaceutical Medical Device Agency in Japan. Classification is 1, 2, 3, and 4. In Australia, it is 1, 2A, 2B, and 3. For Canada, it is the Health Canada Therapeutic Drug Division, and it is classified as 1, 2, 3, and 4. And we'll just now look at the important part, that is medical trial, device trials, clinical investigation or evaluation. The points how we have already seen how a drug is different from a medical device. We'll just see how a drug and a device is different. In healthy subject recruitment, new drugs are studied carefully in phases starting with introduction to a small number of healthy subjects. For medical device, especially those that are required as surgical implants, it is not possible to insert the device in a healthy subject. That is the main difference between a device and a drug trial. Total number of subjects needed to show safety and efficacy is in thousands. In device trial, it's 1 to 200. Use of placebo. Placebo is basically a sugar pill or a dummy drug that looks like a drug but doesn't have its efficacy. For many drug trials, the control is placebo. In medical device studies, it is often unethical to use a placebo. For many devices, it would be impossible. Randomization and blinding, easier to perform. It is very difficult in device. Standard follow-up tools are ECG, BP, HR, biochemical parameters, and devices. It is imaging could be an option for functioning assessment. Necessity required in every case, minute changes of the composition of drug, and necessity not always required, whether or not done will be conducted. Stages in medical device investigation. In drug trials, it is divided into phase 1, 2, and 3, and phase 4 is post marketing. In devices are feasibility, and 2 and 3 are pivotal studies. We'll see the difference in details. Phase 0, it is not always done. It's done in 10 to 15 subjects. Test a very small subtherapeutic dose of a new drug to study its effects and how it works in the human body. Not all the drugs will undergo this phase. In medical device, pilot or early feasibility or first in humans, 10 to 30 subjects are there. Small studies to collect preliminary safety and device performance data in humans, guides devices, modifications, and or future study designs. Phases, safety and toxicity is done in phase 1. Safety and efficacy is in phase 2. It is to phase 1 is in 10 to 100 and phase 2 in hundreds. Phase 1 is true first in human study to test safety and toxicity, usually in healthy humans. Phase 2 assess efficacy and safety in patients. And in medical device, it is equivalent to traditional feasibility that is done in 20 to 30 subjects. Assess safety and efficacy of the near final or final device design in patients. Guides the design of the pivotal study. Phase 3 is clinical effectiveness that is done in 100 to 1000 participants. Confirm clinical efficacy, safety, and adverse effects. Compared to the new drug to standard care or a commonly used drug. Pivotal done in hundreds. Large study to confirm clinical efficacy and safety and risk. Statistically, it is driven. Phase 4 or post marketing is done in thousands. Monitor long term effectiveness and safety in the general population. Post market in thousands. Monitor long term effectiveness, safety, and uses in the general population. Class A, low risk, minimum risk. Clinical investigation is not required. Class B, low to moderate, moderate to high risk, may or may not require. It depends, case to case basis. We look at the pilot and the pivotal study in details. First done in human, intended to acquire specific essential information about a device before going into the pivotal study. Small number of patients and sites, like 10 to 30 patients at the rate 3 to 4 sites. Sites are basically hospitals where clinical investigation is done or clinical trials are done. It is exploratory in nature. The object of a pilot clinical investigation includes assessing feasibility, example, preliminary device performance, exploring eligibility criteria, and their practical application by pivotal control investigation, ascertaining potential harm, Primary safety evaluation, studying device mechanism, validating a method for determining an outcome. Pivotal clinical investigation. Pivotal clinical investigation shall be carried out on the basis of data emerging from pilot clinical investigation. The pivotal clinical investigation is a definitive study in which evidence is gathered to support the safety and effectiveness evaluation of a medical device for its intended use. The study is confirmatory that may be conducted in a large number of patients with disease or condition being studied and scope to provide the effectiveness and adverse effects. For investigational medical device which does not have a predicated device, prior to conduct of pivotal clinical investigation in Indian subject, the Central Licensing Authority, that is the DISCO, Central Drug Standard Control Organization in India, may require the pilot study data to assess whether the pilot data is in conformity to the data already generated outside the country. The data is used for supporting marketing authorization application. Post-marketing clinical investigation is the study other than surveillance performed after marketing approval, which means the medical device is now being marketed. The study may not be considered necessary at the time of medical device approval, but may be required by the Central Licensing Authority for optimizing the intended use of the medical device. Post-marketing clinical investigation includes additional drug-device interaction, safety studies, investigation designed to support use under the approved indication, example mortality or morbidity studies, etc. Subsequent to approval of an investigational medical device, the applicant shall furnish periodic safety update report once they are marketed the drug. Now we'll look at the ethical guidelines for medical device. Medical device in the patient should be also be evaluated for safety. Device should be provided free of cost or if expensive at a feasible reduced rate after the clinical investigation has been completed. Avoid therapeutic misconceptions. Any adverse events, AEs or serious adverse events should be reported within the timelines as per the schedule of a new drug. Here, user error could also be the cause of an AE or an SE. If the participants wants to withdraw from a trial, it may not be possible to remove the internal device. For example, a cardiac stent is there; it cannot be removed from the body. This must be explained to the participant before enrolling her or him. The participants, however, should be allowed to opt out for continuing in the trial without prejudice to his or her ongoing treatment. If feasible, post-trial publication should be emphasized with the sponsor. The duration of follow-up should be long enough to detect late onset adverse reactions, especially when the device is implanted within the body. Now we come to the end of the webinar. For any diploma course or webinar related query, you can visit our website or send us a contact us by an email. We have diploma in clinical research, clinical data management, and pharmacovigilance. We also offer services of webinars or seminars in various institutes, colleges, and hospitals. You can always follow us on Facebook for latest updates and YouTube for all these videos. Thank you for listening. Bye bye.